Hi there! You're about to listen to a vintage episode of the Under the Microscope podcast. While the content is still as relevant and as interesting as when it was recorded, our webpage has changed. You can now find us at thesciencetalk.com/real-scientist-nano. Welcome to Under the Microscope. This series is brought to you by the Real Scientists Nano team. Our goal is to provide a platform where scientists can communicate their work and interact with the public. Hi everyone, today we have with us Kwasi Kwakwa, who is an imaging scientist at European Molecular Biology Laboratory, the European Bioinformatics Institute, close to Cambridge, in the UK. Hi Kwasi, how are you doing?
I'm fine, thank you. How are you doing?
I'm good. Looking forward to learning about your research. So let's start by understanding your work. So could you explain your research to us in super simple words, please?
Okay, I'm an imaging scientist, which is kind of a general term for people who mess around with microscopes and other imaging platforms. Mm-hmm. In my case, what I've done across my career is build microscopes or manage microscope systems that are used for imaging, usually biological samples. So it's usually looking for kind of activity going on inside of cells. Yes. And so the current job is building and managing microscopes at the European Bioinformatics Institute, which is a genetics lab. Mm-hmm. And in my lab, the thing that they're most interested in is being able to look inside of cells that are inside of thin tissue sections, mm-hmm. to see what genes are active in each cell. Mm-hmm. So you take, say, a slice of kidney, and the cells in your kidneys are performing different activities from, say, the cells in your heart. So if you look to see what genes are active in the cells in your kidney, you're going to see a completely different set of genes than you'll see if you look in your kidney. Mm-hmm. Now, generally, the way you would do this is in the old way is by genetic sequencing. So you take a cell, you pull it open, you take the genes out, and you read them through, and you sequence them to see what genes are active in the cell. But that only tells you what is on it, like which genes are active inside of the entire kidney. But suppose the genes on the outside of the cells on the outside of your kidney are doing a different job from the cells in the center of your kidney. You can't tell that by just sequencing the entire kidney, but you can if you take a slice of it, put it on a microscope, stain it so that you have fluorescent molecules that attach to the active genes inside the cells, and then put that in the microscope, and the microscope can go along and read that and tell you which cells and which parts of your kidney are doing what kinds of activity.
Wow, that sounds super cool. So you must have, first of all, super detailed images and super colorful images as well.
Yes, it's good for pretty pictures. It's fine.
Okay, super. So I'm assuming what you mentioned with the kidney slice, it could be basically any tissue slice, it could be any organ slice, it could be basically anything, right?
Yeah. So the lab we have a bunch of collaborations with a bunch of different labs looking at different kinds of human and animal biology. So people who are studying brains or kidneys or endometriums or feet, we look at all of it.
Wow. So there would be a lot of beautiful pictures, which brings me to the next question, that is, what can the followers expect in the week that you will curate the Real Scientist Nano Twitter account?
Well, I'm a microscope person, so yeah, there will be lots of pretty pictures. Mm-hmm. We're going to talk about caveat, I'm not a biologist. Microscope people are sometimes biologists and sometimes physicists. I am a physicist, so mm-hmm. I can talk to you about the physics of looking at biological samples in a nanoscale, complete with pictures.
Right.
And to kind of how you get there from a general physics degree, and kind of what day-to-day life is like working on microscopes.
Okay, so a lot about microscopes. Okay, what more will you be talking about?
Well, I can't have the entire week be about that. So part of the week will be about that. Part of the week will be about, things that I kind of enjoy outside of science. But so I'll talk about what scientists do when they're not in the lab. But I'll also talk about issues around diversity in science and why that is working and why it isn't. Why it isn't being the far more important question.
Okay.
And then if people are interested in careers in science and what possible options are available, hopefully I'll be able to give some advice on that front as well.
Okay, all right. So there's like a foolproof bunch of information coming from you when you will be tweeting from the account. That's wonderful. Looking forward to having you on Real Scientist Nano. Thank you very much for speaking with me.
Thank you very much for having me.
Thank you for listening. To know more about us, please visit our website realscientistsnano.org and follow us on Twitter at realsci_nano.